Thank you. So, hello everybody, I am Henri Ringeard and I will introduce you now the project I've been working on during the last month, which is molecular and morphological identification of polychaetes from the genus Marphysa from two Portuguese coastal systems. So for that project, I was under the supervision of Katia Marques and João Garces from the Aquaculture Research Station of Olhão in the south of Portugal. So first of all, a few general information concerning polychaetes. So polychaetes are annelid worms, which are one of the largest marine taxa with more than 11,000 species described since 1758. They are present in lots of diverse environments, like deep areas in hydrothermal vents, but also in more shallow areas like coral reef, oyster reef, but also salt marshes. We can find them in sandy, muddy, or rocky habitats. For some species and genus, their ecology and biology have been poorly studied, and that lack of knowledge leads to the fact that the taxa is subject to constant modification. So during our project, we were more specifically studying Marphysa sanguinea and the controversy about that species. So it has been first described in 1813 from the south coast of England. It is a burrowing polychaete living in sandy, muddy or rocky habitats, and it has been during a long time considered as a cosmopolitan species due to lots of its presence a bit everywhere in the world, like Europe, South Africa, Australia, Asia, and of course in Portugal. We had to wait until 2013 to wait for a taxonomist to ask the scientific community to perform revision about the previous description, and a few revisions led to the fact that we discovered four new species, which are Marphysa victori from France, Marphysa aegypti from Egypt, and Marphysa chirigota and Marphysa gaditana from the Gulf of Cadiz in Spain, a place which is located close to our sampling area. So why study that species? In Spain and Portugal, this species is intensively harvested by professional and recreational fishermen to fish the sea bream Sparus orata or Diplodocus sargus, but also other fishes. The sales value there have been estimated around 500,000 euros per year in Portugal, but this is only an estimation due to the presence of a parallel black market in those countries. Concerning the ecological point of view, we don't have actually data on population dynamic of the species, so we don't know if the digging harvesting activities impact the population, but what we know is that activities can impact the sediment, and by impacting the sediment you potentially impact the bunting fauna that lives in and the seabirds that feed on the sediments. So at the start of the project we had three different hypotheses. The specimen from Portugal were belonging to Marphysa sanguinea for the first one. The second one was that potentially our specimen from Portugal were finally one of the two species discovered recently in the Gulf of Cadiz, which were Marphysa gaditana and Marphysa chirogata. And the last one was that our specimen will represent a new species endemic from the Portuguese coast. So for the objective of the study, it was to describe and identify the morphological points of interest which could help us to distinguish our specimen from the one described in the literature. And to reinforce that study, we also wanted to use molecular tools in order to obtain DNA sequences to perform barcoding, which is like a good tool for species discrimination. The overall objective of the study was to resolve the uncertainties regarding the identification of this species in Portugal. So for the sampling, 20 specimens were sampled by professional fishermen, and they gave us the specimen at the station of Olhão. So 20 specimens were Ria Formosa lagoon, which is located in the south Portugal. It is a coastal lagoon system protected by a few islands and constituted of lots of channels affected by the tides. It is an important zone for fisheries and aquaculture. The other sampling area was the Sado estuary, which is located in the south of Lisbon. It is like the second largest estuary of Portugal. It is an important economic pole due to the numerous sectors that take place over there. and most of it is actually classified as a nature reserve. So for the morphological observation, the specimen were first relaxed using a 10% formaldehyde solution before being fixed in ethanol. Once they were fixed, we were able to look at their head region and the abdomen using a binocular microscope. We also performed a few measurements, like the length, width ratio at mid-body or the total length. For the smaller anatomical part that we wanted to study, we decided to dissect them and observe them separately. We also had the chance to go to Lisbon, to the IMBRSea station, to use their scanning electron microscope in order to obtain detailed picture of the parapods, which are lateral projection on the side of the animals, and more specifically about the chaetae, which are small hairs composed of chitin. Concerning the molecular analysis, the first step was to extract the DNA from the samples we had. Once that was done, we checked the integrity of the molecule we extracted. We also checked the size of the fragments and the concentration we extracted. We also had to select a few primers for the gene we desired to amplify. So for the PCR, we decided to amplify two genes, the cytochrome oxidase subunit 1, which is the COI gene, which is really important for barcoding, and the second one, the 16S ribosomal RNA, to reinforce our study. So for the COI gene, we used a pair of primers from the literature, which was ACOIAF and COIEU-R. For the 16S gene, after I've been tried with like a few primers from the literature, we decided to design proper ones, that you can see below. Once we obtained our PCR products, we decided to send them to sequence at the CCMR Molecular Biology and Sequencing platform in the University of Faro. And when they send us back the sequence, we use them with some of them extracted from online databases to perform an alignment using the TCOFI tool online and to construct a phylogenetic tree using the software MegaX and the Kiroma two-parameter model with the bootstrap method with 1000 replicates. Concerning the results, so we decided to look at different anatomical points, which are really interesting. The first one was the maxillary apparatus. What you can see on the left is that the maxillary apparatus is constituted of lots of different pairs of mandible and jaws, and the different pairs have different numbers of teeth, depending both on the specimen but also on the species. As you can see on the right, I put the maxillary formulae for the specimen we observed from the Sado estuary, the Ria Formosa, and also the one I extracted from the literature. And you can see that for the maxillary pair 2, 3, and 4, the number differs depending on the species. So it was the first point that was a good difference. Then we decided to look at the pectinate chaetae, which I told you before are small hair composed of chitin on the side of the animal. And as you can see, it exists different types of pectinate chaetae that differ depending on the size of the teeth that you can see here and also of the number of teeth. So for the specimen from Ria Formosa, we were able to observe 4 different types of pectinate chaetae. Only 3 were observed for the one of the Sado estuary. And in the literature, 3 were described for Marphysa gaditana. Finally, the last important point that we observed were the maximum branchial filament observed on the parapods. As you can see on the picture on the right, this is the branchial filaments, and that number varied depending on the species and depending on the position of the segment along the body animal. So we could observe 5 for the Ria Formosa specimen, only 4 for the Sado estuary specimen as maximum, and 6 were described for the description of Marphysa sanguinea. We also had the chance to look at other different anatomical points like the ratio length width, the position and visibility of the eye, but they were like minor points. For the molecular analysis, after the amplification and sequencing, we were able to obtain 3 sequences for the COI gene, which were unfortunately way shorter than the one that the 800 base sequences expected. Hopefully, for the 16S gene, we were able to obtain 6 sequences with a good length. We used all these sequences sequences to construct that you can see on the right. This is the tree for the 16S gene. Unfortunately, after we built the tree for the COI gene, we didn't use it for the study as there was like a clear lack of coherence in its construction. But what we can see for the 16S gene tree is that all our samples are packed together here on the top with no visible distinction between the individual from Ria Formosa with RF and the one from the Sado Estuary with SE. So that was the first point to observe. What we can observe is also that all samples are packed together in a single clade with the one from Marphysa gaditana here in the blue box. And this clade differs from the one from Marphysa sanguinea that is here in the green box. The bootstrap value that you can see here, 64, means that out of 1,000 replicates, we obtain 640 times that simulation, which means that potentially all specimens differ from the one from Marphysa gaditana. It is a significant value, but not a strong value. If we compile all the molecular and morphological results we obtain, we were able to propose a potential new update for the geographical range of Marphysa sanguinea. The limit on the north does not change and remain the nasal and waters, and that is probably due to the environmental condition occurring there, which limit the expansion of the study for Marphysa sanguinea. But for the limit on the south, we can maybe propose a new limit at the north of the Sado estuary, as we see that potentially our specimen does not belong to Marphysa sanguinea. That work is one more confirmation probably that Marphysa sanguinea is not the cosmopolitan species described in the past. So if we compile all our results, we've been able to state on 3 different points. The first one being that our specimen seems to differ from Marphysa sanguinea, both by looking at the molecular results with the 2 groups we can observe here and here, and also by the difference we observe in the morphology, like the pectinate cytae type and the number of branchial filaments. The second point that we can state is that potentially our specimen on each species differing from the one from Marphysa gaditana, as we can see that they form a second group, but this needs require confirmation, more confirmation. Finally, it is impossible right now with our study to make any distinction between the individuals from Sado estuary or from the Ria Formosa, even if we observed a few differences from a morphological point of view, like the overall size during the study. If it was possible to modify a few things in my study, it would be to amplify maybe in a better way the COI gene, or maybe to try to improve the sequence I obtained before, and that I could do it using maybe a new pair of primers, or try to clone the fragment I received after the amplification. I also would like to improve the pictures I did with the scanning electron microscope, but that due to the COVID-19, I hadn't that much time at the station of Lisbon. But I would appreciate the help maybe of a taxonomist for the morphological part, as I think it is like really important, and I have been missing a bit of experience. So what I can do, to conclude, I would say that identify species is clearly necessary for further studies and to consider harvesting organisms in a sustainable way. Once that will be done, it would be interesting to envisage to study their population dynamic and their reproduction, implement a management measure in Portugal by the amplification of COI or by the improving of the extracted. And I would conclude saying that potentially our work observed in Portugal could represent a new species, but this needs further confirmation. I would like to thank my supervisor, Katia Marquez and João Garces from IPMA, the whole team of the aquaculture station of Olhão, the IMBRSea program, and all the university I passed by to give me the chance to study what I like. Thank you.
Okay, thank you.